All right.
Well, welcome everyone to our seminar series on biomedical applications. And today's speaker, our first speaker today, is Dr. Vincent Plugnall from University College London, who is going to talk about copy number variation.
Okay, thank you very much for inviting me. So, I'm aware of the theme of the workshop about change point, the theory. There is some change point is built into what I'm going to talk about. Some of the bits are somewhat trivial from a change point of view. At the end of the talk, the first bit I'm gonna talk about, section 1 and 2, is things I've done some time ago, so it's pretty stable. And again, the change point aspect are pretty understood and not very challenging, but nevertheless, I hope it's gonna interest you. And there is this issue of how we apply those ideas to non-invasive prenatal diagnosis, so how you diagnose chromosomal abnormalities in the fetus before birth. So that's an important medical application of those ideas. And that's where I feel like the way we're gonna use those technologies is not completely sorted. And it's still very early on. It's the work from Kitty Lowe, who worked with me at UCL. But we are in the first steps of making this thing work. And I'd very welcome some feedback on what you think are the good ideas from a mathematical point of view. So, first of all, you already had a talk at the beginning of this workshop about copy number variance. I just want to clarify exactly what I mean by that. For the purpose of this talk, I really mean either deletion or duplication. So you have a big chunk of the chromosome that can be cut out, and you get a shorter chromosome, and you have or duplication that can be quite large. The size can vary extensively. You can have an entire chromosome duplication, Down syndrome being the most famous example of full chromosome duplication. That would be chromosome 21. Happens to be a fairly small chromosome, so we can actually, there are developmental issues, but we can survive with 3 chromosome 21. Few chromosomes can allow that sort of things. You could not survive with 3 chromosome 1, for example. And but the size can really vary. It can be as small as a few kb or something like that. If it removes a key gene or a key exon or it duplicates a key gene, some very bad things can happen. So there is a wide range from a few kilobases to an entire chromosome, which all of them are interesting. And the background, of course, to that is the fact that we've been able to, the cost of sequencing has dropped tremendously. So this graph is being shown in every single talk almost about human genetics. But I mean, I started my PhD around 2002. You can see you used to pay almost $10,000 for a megabyte of sequence. We've gone down to 10p and it keeps decreasing. So that's really the background for why those technologies are becoming more popular and widely used. And one thing I do extensively, that's one of the main focus of my work at UCL, is try to diagnose individuals with very rare genetic disorders. So someone comes to UCL, and there are plenty of hospitals at UCL, people who come with very well-defined disease, whatever that is, heart disease, dermatology, bone marrow failure, whatever it is. And we sequence them and try to identify what is going on in their genome that is causing the disorders. And that technology of exome sequencing has radically changed the way we do that business. So just to get the terminology right, exome sequencing is a cheaper version of whole genome sequencing. We don't sequence the entire exome, but we have an enrichment step that enriches 100-fold or something like that, the bits we sequence for the protein coding part of the genome. So we sequence 1% of the genome, but that's perhaps the most important 1% part of the genome. This process of enrichment introduces a lot of biases, and all those technical biases is something I will talk about a bit later, because they really matter from a mathematical standpoint. So here is an example that's from a paper we published. So I will explain what those graphs mean, but very briefly, you have genes here on this panel here. You have a measure of read count, and I will explain that in a minute. And here you see that there is no read at all, because in that individual, the entire right side of this gene, LRBA, has been removed. Both parents had the deletion, they passed that to the offspring, and therefore this LRBA gene is not working, and that's what's causing a rare disorder in that individual. So for the point of view of Mendelian disorder diagnosis, I'm really talking about those very clear-cut, very definitive, the gene gets removed or completely duplicated sort of situation. That's a medical background for what I'm going to talk about today. And if I may insist, please stop me at any point throughout the talk, right? I don't think I'm going to be too long today, so I'd rather have you stop me during the talks and ask questions at the end. I'd much rather that. So we have the short sequencing reads, and So I want to give you an idea of, perhaps you're familiar with that or perhaps not. So we have those short reads that here. So they're paired end, typically, means that we sequence, it's 1 400 base pair fragment, so that goes from here to here. We sequence one end going from left to right, and the other end typically going from right to left. We always sequence 5' to 3', so the segment going left to right on, on this strand, and we have the other DNA strand we sequence in that direction. And we have plenty of them, so forget about this red region here, but we have millions of those small fragments that are scattered throughout the entire genome. And that's the basic short read sequencing technology that is being used to generate those sequence data. So how do we call copy number variant using sequence data? The principle is very basic. We have this strategy called read depth. So what you see here is a measure of the read that are mapping to a given region. We have a baseline, so it's a paper by, I forget to put the reference, Yoon et al. It was published in Genome Research in 2009. I apologize for that. But the basic idea, you get what's going on. In this situation, they propose to bin the genome in 100 base pair windows. So this binning strategy is mathematically not quite elegant. I mean, it's not very clear that it's the optimum thing to do, but that's what they've done in this first paper, where they show that the technology works. So you look at those bins, you compute a normalized version of the number of reads in that bin. So you have an expectation, you compare that to the expectation. And when you have a drop here, well, it's probably because you don't start with 2 copies of the DNA, but you start only with 1 copy of the DNA. Basically, the number of reads you're getting will be proportional with the number of DNA copies you have in the first place. If you have 3 copies, you get more reads. If you have 1 copy, you get fewer reads. And if you have 2 copies, well, you get the usual 2 chromosomes, the expectation, right along the baseline. And you get those figures that look like that, where you get normal, 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 big gap, and another big gap here. Here, this individual is normal, normal gap, and again, and the deletion that is being observed as this part of the sequence. So is it?
Roughly speaking, what is the size of those copy number variants?
So again, they can vary completely. It can be an entire chromosome, so 50 megabase or something like that, or it can be 20 kb or something. So I'm interested in the broad, the whole range. It's usually fairly straightforward to detect the large ones, but in the context of prenatal diagnosis, you will see that even the large ones can actually be quite challenging. So anything really, we want to find any of those in general, as long as they're rare and potentially disease-causing.
But you're talking on the order of 20 of kilobases, not on the order of bases.
Well, let me go a bit further, and I think the rest of the talk will clarify some of the issues. They can be quite small, but yeah.
You mentioned you were dealing with parent.
Yeah.
So to what extent the fact that it's parent?
So the paired end strategy, I think I removed the slide I wanted to show the matter. So if you get quite lucky and your paired end spans exactly the location of the copy number variant, you can use the distance between the pairs to gather information about the presence of a copy number variant there. In the context of exome sequencing, it's unlikely to be very useful because we only sequence 1% of the genome. So your read would have to fall exactly at the breakpoint of the CNV, which will happen 1% of the time if you sequence 1% of the genome. So all the strategies that are not read depth based, like the one that you have here, are very unlikely to be useful in the context of exome sequencing. They are very useful for whole genome strategy when you have a good depth throughout, but I'm essentially, for the purpose of this talk, ignoring anything that is not read depth based. Actually, I say that, but in some clinically relevant cases, we did identify some copy number variant because of those signature of paired reads. So actually, if you're lucky, and if you look at enough cases, you do find them once in a while, and they're useful. It just, most of the time, this is not the strategy.
And what's the read depth you're typically using?
So again, it depends quite a bit. In this example here, that Yoon et al. Paper, they use a 100 base pair bin, so the read depth has to be very high. You can get actually a picture here of the number of reads mapping a given region. They're talking about the whole genome sequencing at 30x or something like that. With exome sequencing, we have also something like 50x, but just for the exon. You will see that for non-invasive prenatal diagnosis, we're looking at 0.5x, something much more shallow. So let's assume right now we're talking about a typical 30x sort of coverage or something like that. So here is a problem that makes the work challenging with exome data is those graphs are really views of the theoretical view of the data, they don't look at all like that when you work with exome sequence. And the reason why that matters is that when you have exome data, you have this enrichment process that enriches for exons because that's what you care about. The process will work very well for one exon, but not so well for another exon. And your normalized read depth will vary tremendously between one exon and the next. So what I'm showing you here is a measure of the read depth, something normalized for the total number of reads being generated and the size of the exon. Those details don't really matter here. For every single exon in the human genome, you have one sample here, another sample right here. And you see that the scale of this coverage vary between 0 and 250, and even quite a bit more for a few outliers. Meaning some exons are extremely highly covered, and some have a very shallow coverage, just because of the way these enrichment strategies do work in practice. So this won't even look remotely close to that. What you get is up, down. I mean, something completely all over the place because of those, that first step of sequence enrichment. All right. So why this is a good way to think about the problem. In practice, things don't work at all like that. This graph, however, points you to an obvious way of dealing with the problem. Is the fact that when you have two samples generated together, one sample is a very good control for the other. You see that there is a very tight correlation between the read depth for one exon in one sample and the next. So essentially, points that are here or here will be the ones you want to look at when you think there is deletion. This one, for example, is a fairly low for sample 2, but high for sample 1, while you think it's probably being deleted in sample 2. And the exons that are here are probably deleted in sample 1, but present in sample 2. Okay, do you get this idea? So things that are not consistent between one sample and the next is probably because they're deleted in one sample, but not deleted in the other. So this idea is in fact a very well-known idea in the field of exome, in this field. It's something called array CGH. So comparative genomic hybridization arrays do exactly that. They hybridize not one but two samples with a green and a red channel on the same array, and they look at the relative ratio of red and green to see whether there is more of DNA for one sample or the other. So it's been very successful, and we always use that in genomics. When we have something too noisy, we use a control point, a reference point that we can compare things to. And by using this sort of shared reference point, we can see something much better controlled. If there is a noise at one point of the array, say here, it's likely to affect the green and the red channel together. The noise will cancel out, and therefore your signal will become much clearer. And that's the reason why those array CGH technology have been so effective to call copy number variance. And that's exactly what I'm proposing to do here, is the fact that we can use one sample to control for the next. All right? So it's a ratio of the two, which we're going to use in the rest of this talk. So how do we model this comparison? So we have, we combine the reads. We have a test and a reference sample. So the test is the one you want to call your disease. You think there is a disease for the test sample, and you have a reference sample. And reads can be assigned to either of the two. We have a number of reads, the ij, so that the reads that fall at a given exon, the read I, and whether it falls for one exon, for one sample or the next. And we estimate a probability alpha, which will be the overall ratio of read depth between the two samples that tells us what is the probability that a given read will fall for in one sample or the other. So if you have 100 million reads overall, one sample has 40 million, and the other one has 60 million, your alpha will pretty much be 40%, tells you that on average, 40% of the reads will belong to the first sample, and 60% of the reads will belong to the other sample. In practice, there are technical covariates. Those details don't really matter for the point of this talk, but the GC content, for example, can matter quite a bit. So we want to correct for something such as features of the DNA. So perhaps one sample will be more enriched for GC-rich regions than the other, and we want to control for those covariates. So basically, we have a model of some sort, a logistic, like essentially a binomial sort of formulation, and we want to infer the probability for each exon that a random read, will go to the test or to the reference sample. So from a graphical point of view, so just that those are some numbers again. 40 million reads, 60 million reads, alpha would be 60% if what you care about is exome 2. And if you only find 20% of the reads mapping to the test exome instead of 60, well, you think, well, there is something going on here. Maybe you can infer the presence of a deletion. We have too few reads compared to our expectation. So if you want a graphical display of the data, that's how it would look like. I'm taking, again, this idea of one test and a reference sample. We have a read count ratio between the two samples. So you see that the average is 40. So it means that on average, 40% of the reads will be mapping to the test sample, the one who is ill, we want to diagnose the disorder for. And we have, so we have this shape. So when the read depth is very low, you get a lot of noise, which is what you see right here. And as the read depth becomes higher, this binomial noise becomes tighter and tighter, and the proportion of reads that are mapping to a given sample is becoming more tight around the average of 40%. Right? So does this graph make sense? We have this, so each cross is one exon in the genome. So we have 200,000 exons roughly. Some are poorly covered at the bottom, so are very highly covered on the top. But on average, something like 40% of the read for each given exon will be mapping to the one test sample we want to call CNVs for. And things, again, outside of this range, points here perhaps, or points here, who have an excess or too few reads mapping to the test samples will be the one where we think there is a CNV that may be located. Okay? So you can fit a binomial, if we take this very basic model I described before with a binomial sort of approximation of the data, things don't look right at all. So that's a binomial fit for the data, and I took the 99% confidence interval of what this interval is supposed to be. And you find that something in this example, which I've picked, realistic data, which we got from the Beijing Genome Institute, 7.28% of the exons are located outside of the 99% confidence interval. So basic, this rule, 40% of the exons mapping to one sample and 60 to the other, clearly there is something that's not quite right because you would be calling copy number variance for 8% of the sequence, which is not realistic. Those CNVs are quite rare in practice. You may have, maybe 0.5% of CNVs, of your exons affected at the very maximum. I mean, it should be actually quite a bit fewer than that. So clearly, fitting a binomial model to those data doesn't make any sense. So the way we've dealt with that is we use a beta binomial model. So we assume that it's a fairly typical approach to deal with those data. So the number of reads mapping to a given sample would be a binomial term. Nj will be your total read depth at this sample. So Nj would be what you have on the y-axis right here. And you have a probability lambda j, which is this time a random variable that is being sampled from a beta distribution. And this beta distribution has a mean, pj, and over dispersion parameter phi. If phi happens to be 0, well, your lambda j will always be equal to pj, and you revert back to the binomial setting, the basic binomial approach. But if phi is non-zero, you create a bit of noise for this pj parameter, for this lambda j parameter, and therefore you get an extra dispersion that is accounted for when you look at your read count data. All right? So it's a very typical over-dispersed model for binomial data. And when you look at the mass, you can see that the variance is divided into two terms. When the binomial term that we're used to, that's a typical binomial term in 1 over n, that goes to 0 when n goes to infinity. And we have a second term, which is this v beta, which is the over dispersion of the binomial, that does not go to 0. And that's really the key issue of the data, is that if you have noise, you can increase your read depth as much as you will ever want to increase your read depth. It's not going to make a difference. You will never get a very tight distribution. And you see it very clearly in those graphs. As you go higher and higher, you will never get something very tight at the bottom. The fact that you get this width that you cannot get rid of is exactly this beta binomial over dispersion variance that does not go away as you keep increasing the read depth higher up and higher up on this graph. Okay. So this model fits much better the data. So here is what extra binomial variance would look like here. Still not quite right. You see you get some sort of noise at the bottom here, but it's still incredibly better than the basic than the binomial model that you have in red here. The fact that it doesn't quite fit the data as well as we should is important, and we don't have quite a solution for that. We're living with this beta binomial model for now, but it's fairly clear that it will generate some, it's quite likely for some data set at least to generate some false positive. So getting some parameterization of this surface that is exactly fitting the data is a bit tricky to do in the general case. I mean, each data set is a bit different, and we struggle to get something that works all the time. So, and then we have a very natural likelihood framework for that. It's very straightforward to do that. We can have a new P deletion, which would be half of what it should be for the expectation. We have a duplication, Pj, which would be 1.5 times what it should be. That's for 3 copies instead of 2. That's for 1 copy instead of 2. And we can write a likelihood for all those terms. So everything is workable once we have fitted our logistic beta binomial framework. We can estimate the likelihood of the data for the deletion, duplication, normal copy number states for each exon. And we have a model. So for example, if the read depth is supposed to be 10% on average, that would be your confidence interval under the null. That would be what you get for the deletion state. That will be what you get for the normal state. And the whole point is, do you have enough read depth to nicely separate this red area from this black area that you have here? So that's your null, that's your deletion state. And the further apart they are, the better you can separate a deletion from a normal copy number state. And you can see again that you get this sort of a limiting situation where it doesn't go down. I mean, if you go beyond 200 read depths, you don't improve the accuracy of the calling for CNV simply because the beta binomial takes over, right? And the binomial variance becomes negligible compared to beta binomial noise. So that's one first practical conclusion that we came up with most of the data we're dealing with. Unlike what people think, read depth is rarely the limiting factor. Once you are beyond a certain limit, which is around 200 or 300 reads, it doesn't really make any difference to increase the read depth. So it's more the technological issue, that technical noise that is preventing you from calling the data properly. So what we're doing really is conditioning on the read depth. When we take this beta binomial approach, we have a total number of reads, and we see where they map the first or test sample. So you could think of that as a conditional approach on the read depth for each given exon. When we submitted the paper, we had quite some discussion with some of the reviewers who had some different ideas. They were thinking, you should be using a negative binomial approach. And saying, it should really be equivalent to a negative binomial once you do that, once the read depth is sufficient. I never get to the bottom of those arguments. I just don't think that's the case. The nice convenience of the beta binomial approach is that your parameters, this alpha, which is a proportion of reads mapping to the test sample, will be estimated genome-wide. You can use all your data and you have a single parameter to estimate. That's the consequence of this conditioning on the total read depth at a given exon. When you look at the negative binomial description of the data, you have 2 parameters that have to be fitted for every single exon. So you have as many parameters as a number of exons to fit to the data. And I just think it's quite different. I think the estimation works in a very different manner, and the precision with which you estimate your parameter is much cleaner with the conditional on the read depth, the approach conditional on read depth compared to taking a negative binomial that does not do this conditioning. So I do think there is something quite different between the 2 approaches, but I'm not completely comfortable, making definitive conclusions about the differences between the 2. So how does it look in practice? So here is some data. It's a real patient. He actually died 2 years ago following bone marrow transplant, complication from bone marrow transplant. And you have, each cross is an exon, and you have a confidence interval, which is this gray area that you have right here. And here you have a pair of exons next to each other that are outside of the gray confidence interval here. And when you work out the likelihood with a reasonable prior, you get a fairly confident call in that region. It involves a gene called GATA2. And just removing those 2 exons of GATA2 is enough to make the gene non-functional, which leads to a leukemia type phenotype. So this graph can give you an idea of where the change point situation here is located. And it's pretty straightforward. It's because with exome sequence data, you get this very discrete nature of the data. You get those exons one after the next. So the way we combine the likelihood across exons, very often a single exon doesn't give us enough information to call a CNV, but 2 of them does. And we do that in a very naive way using a hidden Markov model. So we have 3 states, deletion, normal, or duplication. And we can jump into one state. And if we go into one state, we tend to stay into that one state. And we pay a likelihood price to jump into the new state. And the HMM does a pretty good work in that case. So it's almost I like the mathematics to be more elegant than a very basic HMM in a sense, especially to this audience. But I really feel like in the context of this very discretized data set here for exome sequence, where you only have data where the exons are located, here, here, and so forth. I'm marking the exons on the bottom panel. The hidden Markov structure of the data is a pretty good way to summarize the information across exons. Okay? Again, because the data are by nature discrete.
Do you account for the difference?
Yeah, so I've thought about that, and maybe some of you will have a good opinion. I'm not right now accounting for the distance between the two, simply because I guess we have to put a prior on the size of a copy number variant, but there are very large ones and there are very small ones. We like the large ones very much, and we like the small one also just as well. So I never could come up with a parameterization of the distance that I was really comfortable with. So I'm not using it right now, but it should be done. I mean, I'm actually asking the user, so it's all coded in an R package called exome devs. I'm asking the users to input the position. I'm just not using it right now. So I should make the extra effort to come up with something reasonable about the distance. But I'd be happy to hear any suggestion on best use that. It's about the prior and what we think the size of the CNV will be, I think. But you're absolutely right. I should do something like that. So now we'll. What time is it first? You have. It is here. Okay, fine. So that's the basics. Like the basic model we're dealing with. And I'll tell you where I think our main contribution is in that field. And I think it's a set of good ideas that really make a difference in practice. So what about this reference exome? We'll always assume that we have a test and a reference exome. And I've argued that if there is a lot of noise, if the test and the reference are not very well matched, this extra binomial variance, this over-dispersion term, will be quite high, simply because there is a lot of differences between the two samples you look at. The binomial will be a poor fit, and you have to put an large extra variance term to account for the fit. So in a sense, you have a trade-off here. It's exactly like an ACGH experiment. You want to find a small subset of exomes that are very nicely matched, very nicely correlated to the test exome you want to look at. So you want, if you go back to this first graph I showed you at the beginning, this graph here, the tighter the correlation here, the easier it will be to call copy number variant outside of those regions, right? So you want a very tight sort of thing here. However, if you have a batch of exomes, some may be very tightly correlated, and some may be quite differently, quite different. So you have a bias variance trade-off. The more you want your reference to be large, to include many samples, because that limits the noise. It's just a binomial noise goes away when you make your reference higher. But if you include samples that are not well correlated, the beta binomial term goes up, right? Because you create a bias, and your test sample is not very well matched to the reference sample. So you have to decide how to optimize this ratio of this extra binomial term, this extra variance due to the beta binomial component of the data. But you have to make the reference large enough to lower the size of the binomial noise. So we really have a fairly typical bias variance trade-off that we're used to dealing with in statistics. So the approach we came up with is that we take the most correlated exome, and we first start with that. Then we take the second most correlated and we add that, and we create a measure of the expected posterior probability for calling a typical deletion. So we have an example, which I think is a 3-exon deletion or something like that. And we say if we average across all the data, given all the samples that we add one after the other, what is the expected posterior probability for calling this typical deletion? And we're gonna stop adding exomes once this posterior probability drops. And so that's like a greedy sort of approach. We have an expectation of detecting the call. And here is how it looks like in practice. We have this posterior distribution that you have right here. And we have the ratio of read depth, which can really be thought as the number of exomes that constitute your reference sample. And the more samples you add at the beginning, things get much better because that's where the binomial noise goes away. So you see that 2, 4, 6, you make your reference grow and things get much more accurate. But you end up reaching some sort of plateau once your reference has 6 or 7 samples into it. And even for some samples, which I suppose have poorly matched exomes, just a small subset of them that are very well correlated, once you make the reference grow, your actually procedure drops quite a bit. You see that you've reached a maximum here, and then it drops. So the algorithm would decide to stop here being the most, the optimum point that maximizes your ability to call a typical deletion in that sample. So that is, I believe, a real contribution from a practical point of view, because it really says that if you run a single sample on one day, you'll never be able to call copy number variant on that sample. You really need to run batches of samples and avoid as much as possible. Don't take a lunch break between the first and the next sample. You have to do them all in one go, minimize the technical artifacts, and that will allow you to reduce the noise and get some those very tight distributions that give you good power to call copy number variants. Okay, and so you don't need hundreds of exomes, but you need at least 6 or 10 or something like that called on the same day with the same batch. Otherwise, things won't be successful. And that really, I think, is the main practical contribution of our exome depth package. And one data set will look completely different from the next. So here is data from the BGI. You see the distribution very tight here. It gets very narrow at the bottom. That's data from the US. That's from Johns Hopkins, I think. And the width that you have at the top is much broader here than what you have over here. And you can see that the gap between the binomial fit in red and the extra binomial variance of the beta binomial model is much higher in this case than in this case right here. So depending on who generated your data, you will get a very different ability to call CNVs from those data. And you should be aware of that before you start running those experiments. Big genome centers tend to do better because they're more automated. They have all the process nicely smooth and worked out. Whereas smaller centers, we tend to introduce more technical noise. I mean, that's my practical experience with the data. So yes, so I'm kind of repeating it. How well does it work? It depends on how closely we can mimic the ACGH idea. Any human intervention in generating data is bad. It creates variance. Humans are notoriously unreliable. Machines is what we want to trust. So that's, and I should mention at this stage, and another question which also maybe you guys may have opinion on that, is something I've also haven't completely sorted out. A competing package to do the same work is a paper by Crum et al. From the Eichler lab, published in 2012 in Genome Research. And the way they do that, they take a lot of read depths from a lot of samples. So they take 30 samples or something like that. And they use a principal component analysis to remove all the technical noise I mentioned before. And the data looks very noisy at the beginning. You can see that this one sample kind of stands out, but it's a lot of mess here. But after doing this PCA type correction of the data, you get something much tighter, and the deletion stands out very nicely, and you can call your duplication in a very obvious manner here. And I have not yet implemented those PCA type steps in the way I run exome depths. Perhaps I should. I'm just worried about the sample size here. I don't really know how those PCA corrections will look like with 5 or 10 samples, which is a pretty practical and typical usage of generating exome data. If you have 10 samples, so as far as I understand, this work by Crum et al. Works quite well, but people like to run it with 40 or 50 samples, a big batch of exomes, which may not be the situation that many people are in when they have 5 samples they run on one given day. So from a user point of view, I think people are happy using exome depth, what we've done, with smaller batches of exomes. And sometimes those SVD, PCA type corrections work very well under a different set of assumptions. My concern is really that those exome capture steps create such complicated, sophisticated sort of noise that I'm really surprised that those SVD type correction can really clean up the data nicely. It's the case here. I don't know how well it works all the time. When you do that business, you always get users that say, oh, for some reason it doesn't work for my data set. Everything is quite a bit different. My idea of matching the best match exome to the test sample may be more reliable in some cases than others, but perhaps we should combine both and use both ideas to really improve power. It's something I haven't found the time to do, but I'd like to look into that if I can find a bit of time in the next months. Okay, and just a last word on that. How many CNVs do we typically find? Something like 200 CNVs per exome, something like that. Two-thirds of them being deletions. Here is the example I gave you before. So we have this nice discrete structure, and we feed this HMM. And we diagnose a lot of patients. Actually, if I have to give one number, we estimate that 5 to 10% of people with genetic disorders coming to the lab are being caused by copy number variants. Based on our data, we diagnose 5 to 10% of them using those CNV coding tools. So it's really important. I mean, you do lose, not 90% of your data, but you lose 10% of your diagnosis if you don't apply those tools. So they do make a difference in practice. All right, so now I'm coming to something more new that we're working on now. As you see, it's pretty rough. We haven't sorted all the issues, but I think it's quite exciting. And there are some change point issues which are a bit more subtle, which I would love to get your opinion on, is the issue of non-invasive prenatal diagnosis. So it's really a life-changing sort of thing. I mean, for those of you who have kids, you know that at least in the UK and most countries, you go through this diagnosis for Down syndrome, and they use your scan, and if you're at risk, they ask you if you want to go through some more invasive technologies to see if your fetus may have some chromosomal abnormalities. And those are grueling procedures. You have a gigantic needle that gets some cells, from the fetus, and the miscarriage rate is high. You can lose your baby 1% of the time, right? So it's a big deal if you have to go through those steps. And many women had to go through that process in the past. So the good news is that we don't need to do that anymore. So people in the field, researchers in the field have worked out that you can find embryonic fetal cells in the blood of the mother. So there is some free-floating DNA. You can sequence the DNA from the mother, the blood of the mother, and you can use a signature from the mother's blood to detect whether there is or not a full chromosome deletion for the fetus. That's a huge difference. It's mostly given on a commercial basis by American companies. It's frustrating, but almost all of the commercial providers are American in that business. But the NHS is trying to roll that out, and we are part of the team that is piloting the use of those technologies. We have Lynn Chitty, my colleague at UCL, is leading the clinical side of things and trying to see how we can apply that in the context of the UK NHS. So it's quite a different situation with exome sequence data. So first, we want to make it cheap, because cost is a major concern of the NHS. Whatever we implement the NHS has to be as cost-effective as possible. So while for the exome sequence, we have something like 50x sequencing over 1% of the genome, the NIPD strategy is very different. We use a very shallow type depth, 0.5x, over 100% of the genome. So the good news is that we don't have those exome capture steps. We don't have this sequence enrichment for the exons that is creating all the biases I was talking about earlier. So much less noise, because we do whole genome sequencing straight through without having to worry about all those technical artifacts I mentioned earlier about some exons being very different. For mostly cost reason, we sequence at a low depth of 0.5x. Maybe in the future we'll go deeper, but for now, that's what is typically sold for those packages of sequencing. But we sequence 100% of the genome. So the total number of reads generated is actually quite similar between exome and NIPD, but the way they are spread out over the genome is completely different. We're looking at two completely different mathematical problems. And the main technical issue here is the fetal fraction. So how much of the fetal DNA do you get in the blood of the mother? And so it's a mixture, mother's blood, and, mother's DNA and fetal DNA. And if the DNA of the fetus is quite high, you will see quite a bit the extra read mapping to chromosome 21, say, if there is a Down syndrome in the fetus. But if there is a very small proportion of fetal DNA, it's going to be very hard to see that, right? So we have a nuisance parameter. For male samples, male fetuses, we can estimate that from Y chromosome reads, because the assumption is that almost any read mapping to the Y chromosome will come from the male. If you have a female fetus, there is no such simple idea, and it's very hard to estimate your fetal fraction. I actually think it's possible, and we have some ideas on how to do that, but they're not at all ready for prime time, so I won't talk about that just yet. So we kind of stuck. We can estimate it from one case. We have to put a prior in the other, but we have to get rid of this nuisance parameter to get a likelihood framework that does make sense. What people do in practice is embarrassingly simple. They compute a z-score where they look at the fraction of reads mapping to a given chromosome, say 21, if you want to call Down syndrome, compared to the rest of the data. So saying we expect 4% of the read mapping to chromosome 21, actually probably less than that, but let's assume 4% for now. If you get 6%, it's too much, and therefore you should get, your z-score will be quite high when you compare your value to the average. So it's really, so you have a large set of data, you compute the sigma from all your samples, and you see whether you're quite, you're way too high or way too low, which would be characteristic of a deletion of a duplication. So compared to what I described above, it's like the CGH idea, but instead of using another sample, we're using the other chromosomes of the same sample as a reference point. When we look at the fraction of reads mapping to chromosome 21, it's exactly like I was describing before, the fraction of reads mapping to the test sample, but this time your reference are all the chromosomes that are not chromosome 21, and your test is just your chromosome 21, right? You could almost think of sequencing 20 chromosomes as sequencing 20 different individuals, if you want to think of it that way. You have 20 independent quantities, and you can use some as a reference for the others, and that is, that is a perfectly fine way to do that. And what does it look in practice? So that's our data, and you can see here that z-score quantity on the left, the axis actually almost very hard to read. So 0, 10, 20, or something, and 30. And you can see that most of the time the z-score is quite high. All those guys have, all those fetuses have Down syndrome. They've been verified. We've gone through the whole birth process, and we've confirmed that. We use some other technology to validate it. And you can see that all the samples on top have a very high fetal fraction of 10%, and the sample of the bottom have something like 2.5%. And obviously our power to call those Down syndrome individuals is much, much lower when the fetal fraction is low, as you would expect. So those PCA correction steps are still very important. I mentioned that many biases go away, but many biases remain. And here is an example of another duplication of chromosome 13. And you see that in the absence of those PCA correction, which we've applied in that case because our sample size is large, we have 300 or 400 samples to deal with, you can see that the red sample as the one with the chromosome 13, they barely stand out in terms of this score. But once we've applied this PCA correction, they really stand out very clearly. So in the context of an IPD, because our sample size is large, because there are very well understood biases, mostly due to GC content, and this chromosome 13 is a complete outlier in terms of GC content, we really get much better power. I mean, it's striking, it's just night and day. Once we've applied those PCA correction, I've mentioned the first part of the talk. So the set of parameters are quite different, but definitely something that needs to be applied in that case. Now, from a change point of view, things are, I think, much more interesting. There is one sample, where we computed 2 megabase bins, and we computed a z-score for each of those 2 megabase bins that we have right here. And this sample actually is interesting. It has a deletion of the first part of the chromosome and a duplication of the second part of the chromosome right here. It's, here is what a complete duplication would look like in green. The normal sample is in black, and this one sample here has this fairly clear deletion here, and this fairly clear duplication on the other side of the chromosome. And we think we know the biological process that can lead to something like that. It's not completely unsurprising, but you see that there is a breakpoint here, and we sure would like to know exactly the breakpoint, especially if it's deletion and normal. Some knowledge of the breakpoint makes a huge difference in practice, right? If you have a gene which you know is key for a lot of developmental processes, the parents may want to make the decision about keeping the baby or not keeping the baby on the basis of knowing exactly what evidence is there that this gene is affected by this copy number variant. How confident can you be about the CNVs overlapping this very specific gene? And I think that's the reason why. And the genes can be 50 kb apart from each other. So as the more accurate we can be, the more effective we'll be in a clinical type setting. So it really does matter, and estimating the boundaries makes really a difference. However, because of low depth sequencing, the uncertainty can be very large. If you think of 0.5x sequencing and a duplication, so 50% increase of reads at a 0.5x read depth, those are 2 megabase bins, and you see it's already fairly not completely obvious to see the deletion here. So there can be considerable uncertainty, which is somewhat incompatible with the sort of precision we would like to deliver to the parents who want to make a decision about their baby. So it's very important to get that right, and the change point aspect is quite important. So how to formalize this problem in a manner that matches the sort of clinical requirements that parents want to hear. So what people often do is not really appropriate for a problem. For NIPD, there is nothing that has been sorted in a very clean manner. There is some work on CNV coding for whole genome, but for exome, it's fairly straightforward due to the discrete feature of the data I mentioned. For whole genome, people do things like dividing the genome into bins, and this UNet-AL paper was doing just that. But then again, a 2 megabase bin, if we would really like to have a 50 KB sort of resolution, or at least some idea of the boundaries at this level, is really ineffective. I mean, choosing the size of the bin is an arbitrary choice. It's not elegant, probably quite inaccurate. So almost all the algorithms, CBS or HMM that comes next, do rely on the presence of those bins, which I think we want to avoid in that case. There is that paper in PNS published in 2011. It's been edited by Tip Shirani, so I guess it's a good stamp of approval of some sort. And you can see, so it's an idea of where you have, actually they use very small bins, I think something like 10 or 50 base pair or something like that. So the bins are almost irrelevant here. They use some merging strategy, so it's a BIC, a Bayesian information criteria, and they try to merge the bins next to each other when there is no reason not to merge them like this. And they go through the iterative processes. For example, here they merge what? They start by merging, I have some text here. They start by merging, what, it's B4, B6, B7. 1 and 2 are being merged here. Then they decide to merge 6 and 7, and so forth. So they go through this iterative process of merging and cutting the genome using some sensible statistics to come up with a segmentation of the data. So that might be the sort of things we may want to look at, because we don't start a very well-defined bin. We really try to decide what the bins are based on what the data are. But I'm still not convinced that it's the right way to think about the problem. So what I would like to see, and what we're trying to think about right now, is it's not really getting a set of CNV calls, because that's not exactly what we want. We want some really confidence interval on the boundaries. You want to answer those questions like what you have here. For a given genomic region, what is the probability that at least some of it is deleted? Here is a gene. The parents will say, how confident are you that this gene is being damaged by this deletion? If it's 99%, maybe we'll get an abortion. If it's 50%, we may think twice about going down that path. So that's the sort of, so just getting set of calls is not good enough. And also, what is a chromosome region that is deleted with 99% probability? What are you sure of? What can you tell us? We're sure all those genes are being damaged, and therefore we can expect that sort of downstream complications for the baby who is about to be born. So my current thinking, and again, I would very much welcome feedback, because we're in the process of setting that up, and again, I don't think it's been sorted out quite yet, is to try to use some sort of Bayesian framework and move the CNV breakpoints using an MCMC type algorithm and retain the posterior distribution of the location of the breakpoints to make a clinical decision. So it's probably doable, but maybe computationally too intensive, maybe it's overkill, maybe we don't need to be that precise. But I think that's the right way to think about that. The choice of prior matters a lot here. For low depths, with low information in large chunks of the genome, because you don't have very many reads here, your decision will not be overwhelmed by the data, and therefore your choice of prior will clearly make a difference in where you think your copy number variant is being located. So so I'm thinking of something like that, where I'm moving a window that is of variable size, and maybe have a deletion here, and trying to move whether it can be here or it can be there, and I would be iteratively moving the left side and the right side of the CNV to try to see where we think it's going to be located. And yeah, so how to set the prior? Is it really worth the effort of setting an MCMC type algorithm? I'm not completely sure yet, but I do believe that this is where we're going to end up to give some really meaningful information. I mean, those two questions I was mentioning before, what are you sure of? And what is the probability that something bad affects this one gene that we know is important for the development of the brain, is really the sort of question which I think we can answer if we have a full posterior distribution of the location of those copy number variants. All right, so that's my last slide. That's my summary. So I've mostly worked on exome data, for which, and I apologize to this audience, the change point sort of formulation of the problem is pretty straightforward. However, the low depth NIPD type of problem is more interesting because binning the data will be ineffective because we don't have any reads in each bin. We have to think of the prior, we have to think of the estimation. And I just don't think there is a good answer to that problem just yet. I'm thinking of a full Bayesian formulation, but if there are tricks that come from this change point field, I would love to hear them. That may save us a lot of computational time and improve the accuracy of what we are trying to do. And that's it. Thank you very much.
Since I would just love that. It's important, I mean, in the first year model, to include some additional extra information as a covariance, say maybe the length, the width of the beans somehow in your model.
Yeah.
Because I've noticed you're using somehow correcting for
GC content.
Which is obvious. But what about some other information?
So
Doesn't make sense.
we did not find. The whole idea of finding a sample that is closely matching the test one is that the way those covariates have an impact will probably be very similar between the test and the reference sample. That's why in our hands, even the GC content doesn't make a big difference. It's critical for the data I've shown you for NIPD, for example, because we don't have this idea of the chromosomes are very different. Okay, so when you compare chromosome 21 to chromosome 13, the GC content of one and the other is very different. And therefore, it's important to correct for GC content. When we use one sample very closely matched to the other sample, the GC content is the same, so somehow those things cancel out, and that's the reason why it doesn't make a big difference. So we did not find covariates that really make a difference for the exome sequence data. If I may add just one thing quickly on, I do feel like I'm missing something for this NIPD problem, because isn't it the most straightforward illustration of what a change point problem is? You have an intensity here. Somewhere along your line, the intensity drops by 50% or less than that because the fetal fraction is uncertain. And then it goes back to normal value somewhere. I do feel like if the change point field has sorted any problem, that must be exactly this one. I mean, there must be, because I tend to look for the biological, bioinformatics type literature, I think the link with the right bit of theory to deal with that problem hasn't been made completely. But I'm sure that from a mathematical standpoint, there is someone must have worked out this problem. I mean, it's completely, if you guys have suggestions on where to look or the right math paper to solve that out, I'd be very grateful because I haven't, I haven't pinpointed the problem.
Oh, any other questions? Yeah. I have a philosophical question. How powerful is a non-invasive prenatal diagnosis? So, are you actually going to have to do an amniocentesis anyway?
No. So the question of power depends on the size of the CNV. So when you look at chromosome 21 of deletion, things look quite good most of the time, and you may only want to do a prenatal diagnosis, an extra bit of amniocentesis if you're around this score for those samples where you're a bit borderline. But if you're here, there is definitely a Down syndrome for your baby. I mean, so there may be a gray area where further work is, further invasive work is relevant. But for a whole chromosome duplication, the problem is fairly straightforward most of the time. However, for small deletions, that's where the problem is really open. And that's exactly where those change point ideas need to be optimized, because a 3 megabase deletion, again, if I show you the graph which we have here, I think that's the best determinant of power if you want a graphical. So there are 2 megabase windows. And that's your baseline, but the confidence interval would be quite high, and that's your actual deletion. This must be a deleted part of the genome, and you see it's not really below the baseline here. The noise is probably, and here you have your duplicated regions, and again, they're a bit above the baseline, but this has definitely duplicated. It's not really different from the background that you have here, right? So you can sort the problem by using a higher depth of sequencing to some extent, and that's what people may end up doing if it becomes cheap enough. But then people will want to call smaller CNVs all the time, right? If you can be very comfortable about a very small CNV, I mean, again, it's a philosophical question, but look at this patient, right? He died following bone marrow transplant. He has 2 exons that are deleted. So from a NIPD point of view, with enough depth and enough, well-done statistics and bioinformatics, we may be able to see that. It's a lethal condition. We know the sort of syndromes associated with deletion of the gene GATA2. I don't think people will want to abort the baby because of something like that, but I guess it's an open question of how far we want to go and how much we want to use that knowledge. It would be interesting to be able to know that from NIPD at least. Can we do it technically? Is a different question from how precise you want to go for very small deletions like that. Right? Where we put the limit is for each person, I suppose, to work out.
I missed something that's important, and I wasn't sure. Well, what I missed was in your use of the beta binomial.
Yeah.
There's a difference between assuming that the binomial is overdispersed with a fixed overdispersion,
Yeah.
and that the individual Bernoulli observations that make up the binomial are overdispersed. Because in the second case, you can drive down the variance by more averaging. And
so each
clear to me why why your formulation was the right one for the science that you're doing.
So the answer is I'm pretty sure that at least from a practical point of view, it fits the data fairly well. So I may have wrongly written the math when I did that.
No, you've written the math correctly, I think.
So why wouldn't it be appropriate? It's just saying that for a given exon, if you look at what it means from a graphical point of view, it means that if you have a simple binomial, it tells you that the mean should always be 40%, and therefore you should be distributed around this mean. When you have this beta binomial approach, it tells you that the mean could be anything between 0.37 or 0.38 or 0.43 or something like that. We have uncertainty about the actual center of the binomial distribution, and therefore if reads are a bit on the left end, it could be because your true underlying binomial happens to be 0.35 instead of 0.4. So your data are still fitting a binomial, but your true parameter of the binomial is moving around.
Right, but now, let me try this. I'm not sure that I can even say, except to repeat what I said before. If the mixture that the beta mixture is applied to the binomial distribution, then you'll get the situation you described.
Yeah.
If the beta mixture is applied to individual Bernoulli observations, and those are repeated, independent observations.
No. It's being applied to each exon independently. So for a given exon, you sample the parameter of the binomial, and then all the reads for that given exon. The parameter of the binomial is exon specific.
But not read specific.
Not read specific, exactly.
You don't draw a new parameter.
I don't draw a new parameter for each read. I draw a parameter for an exon, and all the reads from that exon will go to the test sample with the same probability.
Okay. And why is that scientifically relevant?
Because if an exon, you expect 40% of the read on average to go to the test sample for a specific exon. Okay. If this exon is a bit funny, the GC content is a little bit higher than it should be or a bit lower than it should be, it could be that those technical artifacts make that it's not 40% of the read that will fall to that exon, but 35% of the read or 45% of the read. Yeah. So it's capturing the fact that this exon may not be fully representative of the genome-wide average, exome-wide average, I should say. And it's interesting this wiggle room.
Okay.
Yeah. So thank you for that.
Anything else? Comments, thoughts, questions?
All right, thank you very much.
Yes, please join me to thank